everybody. My name is Joanna Blodauer. Today is January 17th, 2021. Welcome to the presentation of the determination of gender, genes versus hormones. Okay, we'll just start with an overview. Humans are sexually dimorphic. Males and females are physically distinct. Di meaning two. Genes and hormones play a significant role in sex determination. Every cell of the human body that contains a nucleus has 46 chromosomes. And of these 46, this consists of 22 pairs of autosomes and 1 pair of sex chromosomes. This is also called a diploid number because the number of chromosomes are in pairs. So the amount of DNA in a diploid cell can be expressed as 2n. So if you look at this picture, here's all of our 22 autosomes and 1 pair of sex chromosomes. So the male and female sex organs consist of 3 sets of structures. They're gonads, internal genitalia, and external genitalia. These gonads produce gametes, which are the egg or the sperm. In the males, the testes are the gonads that secrete sperm, and in the females, the ovaries are the gonads that produce eggs. Germ cells are the undifferentiated gonadal cells that will either produce eggs or sperm. The internal genitalia are made up of the accessory glands and ducts that connect the gonads with the external environment. And finally, the external genitalia are the external reproductive structures. Interesting, I learned in this class is that in mammals, the default pathway of sexual development is female. So more in detail on chromosomes. So in our body, we have this thing called chromosomes, and the chromosomes is where the genetic information of an organism resides. So as a human, we have what I mentioned, which was 22 pairs of autosomal chromosomes and that 1 pair of sex chromosomes. So those 23 pairs gives us the total of 46 chromosomes, and half of those come from our mother and half of those come from our father. It's totally random selection, but whenever you get an XY pair, you get a male, X coming from the mother and Y coming from the father. And when you get an XX pair, you get a female. One is coming from the mother and the other from the father. So the presence of this X or Y chromosome leads to the development of internal and external sex organs or gonads of a developing embryo. So as a gonad, for example, a male has testes that produce sperm and female has the ovaries that produce ova, and we'll talk about this again. An interesting point is that the X chromosome is larger than the Y. So here's how the X and Y chromosomes aid in sex determination. This process is completely random. And as you can see by these charts, we have a female with an XX combination and a male with an XY combination. Now each sperm a male produces will either have the X or the Y to pass on to the offspring, and each egg a female parent passes on to its offspring will either can be an X. So you can see down on this chart, an X and X come together to make another female, or an X from the mother and a Y from the father will come to make a male. So the offspring will randomly acquire 1 chromosome from each parent. So if you get XX female, XY pair, you get a male. Moving on to the early stage of development. So this is interesting too. The sex of the embryo is difficult to determine because reproductive structures appear similar to each other in early development. Differentiation can begin to be determined after the 7th week of development. So as you can see in this picture, there are bipotential gonads, meaning it cannot be morphologically identified as a male or female, but it has already been given an X or Y chromosome and it's already received its 2 chromosomes. So now we know it's already decided if it's a male or female, we just can't see it yet. So there's a medulla, which is the center, and then the outer cortex. So if this is a male, then the medulla is going to develop into the testes, and then that medulla will regress in females. And if it's a female, the outer cortex will develop into ovarian tissue, and then the medulla will regress. So as you can also see this tubular structure in this picture, and that's the Wolffian duct. And in males, that's going to stay there, and it will form into the epididymis, the vas deferens, and the seminal vesicles, as I will go into more. And then the tubular structure on the other side is a Müllerian duct. And then in females, that will become the fallopian tubes, uterus, cervix, and upper half of the vagina. So as the growing embryo continues into the next stage, one set of ducts will diminish, as we had said, depending on the gender, and then the other one will stay. So if it's a male, on the Y chromosome of a male, there's the presence of the SRY gene. And I'm going to go into more detail in a couple slides. And that means the sex-determining region of the Y chromosome. And this will stimulate the bipotential gonad into turning into the testes. The SRY gene produces a hormone called the TDF, or testes determining factor. And that's going to bind to DNA and activate all other types of genes to aid in this male development. And the protein products of these genes direct the development of that gonadal medulla that we had just seen into the testes themselves. Once differentiated, the testes will secrete hormones, one being AMH and the other androgens. And of the androgens, we have testosterone and ADH, and both will influence the development of internal and external male genitalia. So an important note to take from this slide is that the testicular development does not require the male sex hormone testosterone. And that's because the developing embryo cannot secrete testosterone until the gonads have differentiated into the testes. More on the male. So the AMH, which is the anti-Müllerian hormone, is a glycoprotein secreted by the Sertoli cells, and that's going to cause the Müllerian ducts to regress. And remember how we mentioned how the Wolffian ducts remain if it's a male and the Müllerian ducts remain if it's a female. In this case, we're talking about a male, so the Müllerian ducts are going to regress. So then we have the androgens, and those are hormones secreted by the interstitial or the Leydig cells, the first one being testosterone. This is going to convert those Wolffian ducts to the male accessory structures, the epididymis, vas deferens, and seminal vesicle. And then the DHT, which is dihydrotestosterone, is going to aid in the differentiation of the external male genitalia. Okay, now I mentioned I'm going to go into this. Here we are. The SRY gene is extremely important, as indicated by bullet point 1. So you can see why that name is called SRY, because as bolded here, sex determining region of the Y chromosome. Tricky. So the SRY gene will only be present if there is a Y chromosome, which means male in this situation. So the presence of SRY gene changes bipotential gonads into the male testes, and also with that presence, there's going to be signal proteins and hormones released in order to aid in the development of the male genitalia. The SRY gene produces the TDF, as I mentioned in the last slide, which will then bind to the DNA and activate all the other genes that produce the proteins to direct the development of the medulla into the testes. And as I mentioned also before, the testes development does not require testosterone. Who knew, really? But after the testes differentiate, they will secrete anti-Müllerian hormone, AMH, from the Sertoli cells, and they will also secrete androgens, testosterone, and the more potent version, DHT, which is produced when 5-alpha reductase converts testosterone into DHT, and those are secreted from the interstitial cells, or those Leydig cells. And so then the testosterone and the DHT are the dominant steroid hormones in males. AMH causes Müllerian ducts to regress, while testosterone causes the Wolffian ducts to become the male accessory structures that I mentioned. Testosterone will control the testes lowering into abdomen to the scrotum, from the abdomen into the scrotum. And then the DHT controls the rest of the male sex characteristics as well as that external genitalia. Okay, let's move on. So you get a female if you get the combination of XX chromosomes. Once an embryo develops, permanent chromosome will be inactivated in each cell in the body and then condensed into a clump of nuclear chromatin, which is called the Barr body, BARR. Now it's totally random, just like a lot of things in this process, and the X that condenses can either be from the mother or the father. You can't distinguish which one comes from where when there's two X's. So therefore, the one that condenses, we don't know if that came from the mother or the father. Also very interesting. So you're only going to get a female without the Y chromosome and in the absence of the SRY gene. The inner cortex of the bipedal gonad will develop into ovarian tissue. And in the absence of the testicular AMH, the Müllerian ducts will develop into the upper portion of the vagina, uterus, and fallopian tubes. So without those male androgens, the Wolffian ducts degenerate and the external genitalia take on female characteristics. being that testosterone is going to influence the Wolffian ducts to degenerate, and the DHT is going to influence the genitalia to take on those female characteristics. So as you can see, it all goes back to the chromosome, which is randomly selected at contraception. And with that Y present, you have the SRY gene, which then influences all these proteins and hormones and genes present to be secreted and cause male development. Only in their absence, with no Y chromosome, a female will develop. And as you can see, the male development seems a little bit more complicated than the female, just now we're looking for the absence of those genes. All right, and this is a fancy transition slide. So it's not the number of gonosomes that is decisive for the gender, but rather the presence or absence of that Y chromosome, as we discussed. And you can see this in the table. And it's clear that the information encoded on that Y chromosome is not enough to guide the formation of such a complicated organ as the testicles, but that SRY gene, which operates really early in the development as a guide or master gene. It has testes determining effect on the indifferent gonads. And the small gene, which is localized on the shorter arm of the Y chromosome, gets expressed in the precursors for supporting cells, Sertoli. It controls a whole number of further genes on the autosomes as well as on the X chromosome. So if you have some extra time in your day and you want to take a look at this table, you can see the karyotype, the phenotypical gender, the gonad, the different syndromes associated with it, and the fate, whether it's like normal development or maybe there's atrophy of the ovaries. It's interesting to go into a little bit more detail because detail is key. X and Y variations are known medically as sex chromosome aneuploidy, or SCA. And I personally found a lot of websites that were focused on this. SCA involve variations in the typical number and type of sex chromosomes. So as we said, the typical number of chromosomes in each human cell is 46, which includes the 24 pairs of autosomes, which refers to all chromosomes that are not the sex chromosomes. And a pair of sex chromosomes, which is either that X or Y, which we talked about ad nauseum. So this means the genetic signature for a person with 46 chromosomes is either 46XY males or 46XX females. So any variation from this typical numbers is referred to scientifically as aneuploidy. So if there's one additional X or Y, it's known as trisomy. And I think you've probably heard of that. Tri denoting the number 3. And genetic conditions that reflect a trisomy include, but are not limited to, Klinefelter syndrome, or trisomy X. And trisomies are the most common X and Y aneuploid conditions. Now, less common are tetrasomies, which denote the presence of 4 sex chromosomes, as you can see here. Still rarer are the pentasomies, indicating 5 sex chromosomes. Typically, 48 and 49 chromosome variations produce more pronounced symptoms than trisomies, although this is not always the case. At the other end of the spectrum lie the monosomies, meaning a person born with only 1 sex chromosome. The only survivable monosomy, SCA, is 45X, and this is a female with only 1 X chromosome, and that's a condition known as Turner's syndrome. A fetus with only 1 Y chromosome and no X chromosome is not viable and cannot survive. By the way, Turner syndrome advocacy and support can be found at www.turnersyndrome.org. About 20% of persons with sex chromosome aneuploidies exhibit mosaicism, meaning they have 2 or more cell lines with different genetic signatures. Sex chromosome aneuploidy is not inherited. The extra chromosome is the result of an error that occurs in cell division when the egg or sperm is formed, or occasionally during cell division after fertilization. An extra X can be contributed by either the mother or the father, but only the father can contribute an extra Y chromosome. Anyway, I thought these were very interesting to discuss. These are my citations. Thank you so much for watching this presentation.